Into details of very interesting lecture that is likely to be delivered by Andres on the topic challenges and practical problems of plant hormones in biotechnology. So, Andres, you are again welcome to deliver your new lecture. Thank you.
Thank you very much. I hope that lecture will be interested. Yep. But I hope that after lunch you have more power to sit and hear my lecture. The subject, of course, will be challenges and practical problems of plant hormone in, maybe hormones in biotechnology. So I will welcome again. Let's start, of course, as biotechnology advances, we have gained incredible insight into phytohormones applications. Of course, from tissue culture and genetics engineering to crop improvement and post-harvest technology. However, the journey is not without its challenges. The practical application of plant hormones in biotechnology face numerous challenges as difficulties in large-scale production, instability under environmental conditions, and of regulatory concern. Additionally, hormonal crosstalk, the complex of interactions between different hormones makes precise control difficult. These challenges demand innovative solutions that integrate biotechnology, nanotechnology, and advances analytical techniques. In this lecture, I would like to present some other waste contaminants dangerous for us, for our planet. That means nanoparticles, micro and nanoplastic. Of course, with correlation to plant hormones. Let's begin this fascinating journey in the world of plant hormones again, and now biotechnology. We know that phytohormones are chemical messengers of plants orchestrating growth, differentiation, and response of environmental stimuli. In biotechnology, they significantly cannot overstand it. They serve as key drivers in several crucial fields. That means tissue culture and micropropagation, where phytohormones are essential in callus induction, organogenesis, somatic embryogenesis, the core techniques of plant tissues culture. By carefully manipulating hormones like auxin and cytokinins, we can regenerate entire plants from a single cell, enabling large-scale propagation of available plant species. The next, of course, key is genetics engineering. It means that modern biotechnology allows us to alter plant hormone biosynthetic pathways, leading to crop with enhanced growth, improved stress tolerance, and high yields. By targeting genes responsible for hormone production, we can design plants that are more resilient to environmental stresses. And third, crop improvement. Through hormone regulation, we can increase productivity and stress resistant in crop. For example, gibberellins can boost plant hay and seed germination, while abscisic acid helps plants survive extreme drought conditions. The next secondary metabolite production were many medical for industrial plants rely on phytohormones to synthesize bioactive compounds. By fine-tune hormone levels, we can enhance the production of available secondary metabolites used in pharmaceuticals, cosmetics, and food industries. So, post-harvest technology, even after harvesting, phytohormones continue to play a role. They regulate ripening, senescence, storage, quality, allows us extend shelf life, reduce food waste, and improve market value. This application illustrates that how hormones bridge the gap between fundamental plant physiology, plant biology, plant biochemistry, and applied biotechnology. However, while their potential is enormous, their practical application is met with significant challenges, from production to stability issues to regulatory concern and unintended physiological effects. The next slide is, I suggest, I would like, maybe not suggest, I would like to omit, because sometimes I talk and show these challenges, more knowledge about phytohormones, which I present in the previous some lectures. I would like to show that the modern environmental threats include nanoparticles, microplastics, and nanoplastics which persist in ecosystem, disturb soil, water quality, and pose risk to plant, animal, and human health. These contaminants can alter plant growth, interfere with nutrients uptake, and affect hormonal signaling pathways. Phytohormones offer a potential solution by enhancing resilience against these pollutants. They regulate stress response similar like other contaminants, improve nutrients absorption, mitigate the toxic effects of these very dangerous and newest contaminants. Additionally, research onto phytohormone-based strategies such as hormone-assisted phytoremediation aim to enhance plant detoxification mechanism, providing a sustainable approach to tackling pollution. Our understanding that hormone nanoparticle or micro-nano plastic interactions grow, phytohormones may become a key important tool in reducing environmental damage and promoting sustainable agricultural practice in the world increasingly affected by these pollutants. We know that nanoparticles are materials with at least one dimension between 1 and 100 nanometers. But why are nanoparticles relevant to plant biology and phytohormones? Their small size allows them to interact with plant cells at the molecular levels, influencing growth, stress response, and hormone regulation. Some nanoparticles can mimic phytohormones, while others can enhance hormone delivery, stability, and efficiency in plants. We must know that, for example, ions, metal, can convert into nanoparticles. For example, silver ions can convert into silver nanoparticles in Chlorella vulgaris, which was a part of my team study. However, despite their potential, not all nanoparticles interacts are beneficial. Some nanoparticles accumulate in plant tissues, potentially leading to the toxicity and give oxidative stress and hormonal imbalance. Nanoparticles can be categorized on their composition and origin. Each type will exhibit distinct properties that influence their application in biotechnology, medicine, and agriculture. For example, there are metallic nanoparticles, metal oxide nanoparticles, carbon-based nanoparticles, and polymeric nanoparticles. Use, for example, are biodegradable nanocarriers made from natural or synthetic polymers. They usually used for controlled release of nutrients, pesticides, and phytohormones, ensuring long-term efficiency in minimal environmental impact. We know that nanoparticles can origin from both natural and anthropogenic sources, with natural source often resulting from natural processes, anthropogenic sources stemming from human activities. For example, natural sources. Nanoparticles can be formed from volcanic eruption, forest fires, and soil erosion. Anthropogenic sources are following: from factories, vehicles, and house, and agricultural application are significant contributors to these nanoparticles. Agricultural practice, such as use of nano-fertilizers and nano-pesticides, contribute to increase presence of nanoparticles in soil and crops and water. Plastic products, as they break down, release microplastic, nanoplastic. It's dangerous, though. This sources highlight that nanoparticles are not just a byproduct of industry's process, but also an inevitable presence in nature. Nanoparticles have shown significant promise in several key areas of agriculture, with application ranging from enhanced nutrient delivery to enhanced pest control and even genetics formation. For example, nano-fertilizers for enhanced plant growth. As example, zinc oxide, iron oxide, or titanium dioxide serve as carriers for micronutrients, enhancing their bioavailability and reducing nutrients leaching. The advantage of using nanoparticles lies in the slow release properties, which prevent nutrients loss and enhance uptake efficiency over time. For example, zinc oxide nanoparticles can improve zinc uptake, which is essential for immune responses. How they contribute to agricultural biology as nanoparticles and nano-herbicide. They can also use to control release of pesticide and herbicide, reducing toxicity and minimizing environmental conditions. For example, silver nanoparticles exhibit antimicrobial properties, preventing fungal and bacterial infection in crops. We know that silver nanoparticles may exist in deodorant, in perfume. stop the odor. Chitosan-based nanoherbicides selectively target weed growth, offering a more precise and environmental friendly approach compared to the traditional herbicide. This example illustrates that potential of nanoparticles to improve both crop productivity and environmental sustainability. However, as with any innovation, there are risks and challenges. For example, long-term accumulation of nanoparticles in the soil, their interaction with plants, and their potential toxicity to beneficial soil microbes remain concerned that need careful evaluation. How nanoparticles interact with soil, water, and air. Understanding this interaction is important for assessing the long-term sustainability of using nanoparticles in agricultural biotechnology. Nanoparticles can have a significant influence on soil structure and nutrient cycling. For example, they can improve water retention, which is particularly important in drought-prone areas. On the other hand, they can also alter microbial communities in the soil, potentially affecting plant growth and soil fertility. In some cases, nanoparticles may accumulate in the soil, leading to toxicity for soil-dwelling organisms. This disruption can have cascading effects on nutrient cycling, which in turn may affect plant health and productivity. In water, nanoparticles, especially those derived from nanopesticides and fertilizers, can leach into water system, potentially contaminating rivers, lakes, and groundwater. Why some nanoparticles, such as iron oxide nanoparticles, have been explored for their ability to remove contaminants from waters, and others may have harmful effects on aquatic organisms. For example, silver nanoparticles can exhibit toxicity on aquatic organisms. For example, Chlorella vulgaris algae, which I studied. Furthermore, nanoparticles in water system may bioaccumulate in this organism, in aquatic organism, plant, of course, in animals, eventually entering the food chain, which can lead to human health conditions. What about in air? Nanoparticles are also present in atmospheric aerosols, contributing air pollutions. They can travel long distance through the air and settle on soil, plants, and water bodies of many organisms, where they can have various effects on plant growth and, of course, ecosystem balance. Some studies suggest that the airborne nanoparticles can interfere with plant photosynthesis and growth, especially if they accumulate on plant surface, blocking the stomatal pores and preventing gas exchange. While nanoparticles offer tremendous potential in agriculture, their presence in environmental raises critical concern. How nanoparticles interact with plants? The most common pathways for nanoparticles to enter is through root or absorption. Often absorbed Nanoparticles travel via the xylem to the shoots and the leaves. The nanoparticles are delivering essential nutrients such as zinc or iron, or if they're enhancing plant resilience to stress. Example, iron oxide nanoparticles can improve iron uptake, which is crucial for photosynthesis and overall plant health. Nanoparticles can also be absorbed through the foliage, through the stomata and cuticular layers. This route of uptake is particularly used for the delivery of phytohormones or other bioactive molecules directly to the plant tissues. Titanium dioxide nanoparticles have been studied to the ability to enhance photosynthetic efficiency when applied to leaf surface. They can be beneficial under high light stress, where they help optimize light absorption and reduce photooxidative damage. Some nanoparticles are known to modulate plant hormone signaling pathways, particularly those related to abiotic stress tolerance. For example, silicon on iron oxide nanoparticles have been shown to prove plant growth tolerance to drought and salinity by enhancing abscisic acid signaling and reactive oxygen species scavenging. Nanoparticles have been a significant impact, such as positive and negative on various physiological processes. The first, of course, the positive effects on plant physiology. Nanoparticles, especially zinc oxide or iron oxide, serve as carriers for essential nutrients, improving their ability and absorbing by plant roots. Nanoparticles like silicon nanoparticles and iron oxide may enhance plant resilience against abiotic stresses. They help by manipulating plant hormones, for example, abscisic acid, which play, of course, a crucial role with drought tolerance and stomatal regulation, on which you better know. Some nanoparticles, like gold nanoparticles, can promote cell division and root elongation by interacting with cytokinin pathways. They make useful in application like tissue culture or plant regeneration. The negative effects on plant physiology. The high concentration of nanoparticles can generate reactive oxygen species, leading to oxidative stress, on which I talk about this. The stress damages cell membranes, protein, and DNA. Some nanoparticles can disturb normal plant hormonal regulation. For example, silver nanoparticles have been found to interfere with oxygen synthesis, leading to reduced root elongation. It similarly act zinc oxide with gibberellin biosynthesis, resulting to stagnant growth. High concentration of nanoparticles, especially those that accumulate on a leaf surface, can block stomatal pores, interfering with gas exchange and photosynthesis. As you can see, nanoparticles have a potential to both enhance and impair plant physiology, depending on factors such as concentration, type of nanoparticles, and the plant species being studied. This underscores the importance of carefully managing nanoparticles application to avoid potential toxicity and environmental harm. How nanoparticles affects on plant tissue and plant tissue cultures and genetic engineering. One of the one of the major challenges in genetic engineering. Of course, nanoparticles such as gold nanoparticles or carbon nanotubes have been used as a carrier for DNA and RNA molecules. And these nanoparticles can penetrate plant cell wall more easily and facilitate the uptake of genetic materials into plant cells, enhancing transformation efficiency. The use of nanoparticles allows non-vital gene delivery, which is often safer and more efficient than traditional methods such as agrobacterium-mediated transformation on gene guns. This is especially important when working with special species that are difficult to transform or even more precise control methods of genetics modification is still required. The next polygylation, pegylation, is a process of both covalent and non-covalent attachment or amalgamation of polyethylene glycol, and silicon atom particles are another example. This nanoparticle has been shown to improve the efficiency of CRISPR-Cas9 gene editing system, enabling more precise and targeting modification in plants. On the other hand, magnetic nanoparticles are also gaining attention for their ability to assist in vitro DNA uptake during plant tissue engineering. The use of magnetic fields can help guide nanoparticles directly into plant cells, making gene editing faster and more precise. The interaction of nanoparticles and phytohormones can either enhance or inhibit the plant's natural responses. For example, silver nanoparticles can enhance salicylic acid-mediated defense mechanism, improving the plant's ability to resist fungal and bacterial infections. This is particularly useful in combining plant disease, as salicylic acid plays a critical role in systematic acquired resistant SIR. Titanium dioxide nanoparticles can increase photosynthetic efficiency by enhancing cytokinin activity, leading to improved cell division and shoot regeneration, and this results in increased plant growth under certain conditions. Silicon nanoparticles have been shown to modulate abscisic acid signaling, improving plant ability to tolerance salinity and drought stress. Crosstalk between nanoparticles and phytohormones highlights the delicate balance a plant must maintain to regulate their growth and response to environmental stresses. While nanoparticles can boost stress resistance and enhance growth, they can also disturb hormonal pathways, leading to unexpected physiological effects. Is of course dangerous. It's equally important to consider the impact on human and animal health, especially when nanoparticles are used in agricultural practices and can enter the food chain. For example, inhaled nanoparticles may cause pulmonary inflammation leading to lung disease, such as asthma. Some nanoparticles, due to their small size, have the potential to cross the blood-brain barrier, possibly affecting neural function and leading to cognitive impairments. Nanoparticles such as zero-valent iron nanoparticles are heavily effective at removing heavy metals like arsenic, lead, cadmium from contaminated soil and groundwater. These nanoparticles are the source pollutants and facilitate chemical reactions, then neutralize them. Graphene oxide nanoparticles have been shown to remove organic pollutants such as pesticides or pharmaceuticals from wastewater by acting as adsorbent, binding to harmful substances and reducing their bioavailability. Nanoparticles play essential role in improving water purification process and application of nanoparticles for pollution cleanup presents on opportunity to restore ecosystem affected by industrial pollution, agricultural runoff, and mining activities. However, it's important to remember that while nanoparticles can enhance remediation efforts, the long-term environmental impact must be studied. Now, I would like to shift our focus on very promising development in the field of nanotechnology, the use of biodegradable nanoparticles, which I would like to underline, in eco-friendly application. As we continue to face growing concern about the environmental impact of traditional nanoparticles, there is increasing interest in using biodegradable alternatives that breaks down naturally, reducing the long-term presence in our ecosystem. These nanoparticles have made from materials like chitosan, alginate, and cellulose, represent a major advancement in sustainability. Those nanoparticles are naturally degradable, meaning they break down into harmless byproducts over time, ensuring that they do not accumulate in the environment, what is once again, very important. For example, chitosan nanoparticles, which are derived from crustacean shells, have gained attention for their biodegradable nature and antibacterial, antimicrobial properties, making them very useful in agricultural application as biopesticides. The next herbivores, such as insects, can ingest nanoparticles when they mosquitoes. I forget spray repellent. Okay. The herbivores can ingest nanoparticles when they feed on contaminants plants. This can affect the growth, metabolism, and immune system. And nanoparticle can also bioaccumulate in higher trophic levels, meaning that predators who consume contaminated herbivores may experience higher concentration of nanoparticles in their bodies as a phenomenon known as magnification. Biomagnification, of course. This process can disturb ecosystem stability and lead to potential health problem for animals and for us. At the top, the food change and the top change, food change. The long-term persistence of nanoparticles in the environmental raises questions about their potential impact on the ecological balance. Over time, accumulation in soil, water, and plants can disturb an entire ecosystem, affecting microbial population, plant diversity, and animal health. In particular, nanoparticles may have unknown ecological consequences, as the long-term effects are still not fully understood. This calls for careful monitoring and regulation of nanoparticles use in agricultural and environmental application. As I remember, I previously presented information concerning the interactions of phytohormones with nanoparticles. So I will skip some slides, but I would like to summarize. One of the most promising benefits, of course, skip a few slides according to your printer version of my presentation. You have my printed version, I hope. No? But I present the same or similar information about interaction of phytohormones. It will be borrowed for you and not me, but for you. Okay, one of the most promising benefits is a promotion of plant growth through the interactions between nanoparticles and phytohormones. For example, nanoparticles can influence hormonal pathways that can regulate auxin, cytokinin, gibberellin levels. Example, silicon nanoparticles can enhance root elongation via auxin up regulation, and gold nanoparticle can stimulate root regeneration by enhancing cytokinin activity. This interaction helps accelerate plant development, making nanoparticles an ideal tool for improving crop production. Another important benefit is the ability of nanoparticle to enhance plant resilience under stress conditions. We know that nanoparticle can modulate the signaling of abscisic acid, salicylic acid, which are involved in stress responses. Of course, that while a nanoparticle can enhance plant growth, excessive exposure can lead to hormonal imbalances. High concentration of nanoparticles can disturb the synthesis and transport of phytohormones, leading to abnormal growth patterns or toxicity. Yes, there's. Oh, the interaction between presents both exciting opportunities and significant challenges. By carefully controlling the dose, the type of nanoparticles and specific phytohormones involved, we can harness the positive effects on plant growth. So as we can see in table, nanoparticle can enhance the effects of phytohormones, and nanoparticles improve the plant response to abiotic stressors. Nanoparticles have been found to enhance plant immunity, particularly through the mediation of salicylic acid and osmonic acid, offering improved resistance against pathogens. However, they use the high concentration of metal-based nanoparticles may result in oxidative stress and cell toxicity, which could counteract these beneficial effects. Smart nano fertilizers hold the potential for reducing agrochemical waste, and the target delivery of nanoparticle can improve crop yield with fewer inputs, benefiting food production efficiency. As I conclude my presentation on the interactions of nanoparticles and phytohormones in biotechnology, yep, many is
Reprint? Mosquito reprint you need?
no. Reprint?
You need mosquito reprint?
No, reprint, no, I can't. No,
Yeah, that's
I
right.
have.
Okay, just see.
Yeah.
Okay,
Mark,
just go.
Mark.
Go on. Okay.
I bought very strong repellent against mosquitoes, especially tropical mosquitoes. And I can use this repellent, this muga, for domestic mosquitoes. I killed. They are dangerous for me because I'm afraid Plasmodium vivax. So I must every day take special tabs against this pathogen. Yeah. Okay. What about long-term studies, which are essential for understanding the bioaccumulation of nanoparticles in plant tissues and their potential, of course, effects? Some challenges, of course, I would like to present. Researchers must focus on how nanoparticles move through the food chain and the long-term impact they could have on the soil health, water system, and organisms that really on plant for food. The studies will help us identify safe concentration and application methods to minimize risk to human health and the environment. In conclusion, while nanoparticles offer immense potential to improve agricultural productivity and plant resilience, it is crucial to carefully manage their application to avoid any unintended consequences. By pursuing biodegradable options of nanoparticles, optimizing nanoparticles, nanoformulations, conducting in-depth studies and complementing strict regulatory frameworks, we can maximize the benefits of nanoparticles while minimizing their risk. The next challenge which we can find in environment are nano or micro or nano plastics. Much like nanoparticles, have raised growing concerns due to their widespread presence in the environment and then potential to affect plant growth, development and stress responses. Again, the same. Microplastics, typically defined as plastics particle ranging from 1 micrometer to 5 millimeters in size, and nanoplastic, which are even smaller and range from 1 nanometer to 100 nanometers, have become pervasive in our ecosystem. The sources of these particles can be divided into main categories of primary sources and secondary sources. The primary sources refer to plastic that are intentionally produced at smaller and the smallest scales. For instance, we often encounter micro beads used in personal care products such as toothpaste and facial scrubs. These tiny particles, through effects in their intended use, contribute to the growing plastic pollution problem when wasted down drains. Secondary sources of microplastics and nanoplastics are even more concerning, as they originate from the breakdown the larger plastics item. Over time, every day, plastic products, whether from plastic bags, bottles, and packaging, degrade due to factors like ultraviolet radiation, mechanical abrasion, and microbial activity. These larger plastic materials break into progressively smaller pieces until they became micro and nanoplastics, dispersing widely throughout the environment. One of the most significant concerns is contamination of soil. As microplastic and nanoplastic enter the soil, they affect soil structure and water retentions. They can disturb essentially soil microbial communities, which play a crucial role in the nutrients cycling, soil fertility, and plant growth. Microplastic and nanoplastic also pose a significant threat to water quality. When plastic particles enter rivers, lakes, and oceans, they can be ingested by aquatic organisms, including fish, marine invertebrates, plants, and others, causing physical harm or by accumulation of toxins. This plastic can also disturb marine ecosystem by clogging the respiratory system of aquatic organisms, reducing their ability to absorb oxygen. One of the most critical impact of microplastic is on microbial communities and soil fauna, both which are vital for the health ecosystem and sustainability of agricultural systems. We know that microplastic enter the food chain through several pathways. So I would like to go. microplastic or microplastic and nanoplastic interfere can alter, of course, hormonal pathways in plants. They interfere with the transport of auxin and cytokinin, which cytokinin stop. Additionally, for example, polystyrene nanoplastics disturb abscisic acid regulation, and plastics have ability to absorb phytohormones, which reduce their viability in the soils. A case in this point is abscisic acid, which binds to microplastic particles, those becoming less available for plants to respond to drought stress. The interaction complicates the way plants manage environmental challenges, emphasizing the need to understand this mechanism better for efficient agricultural practice. Microplastic and nanoplastic are not just passive pollutants. They act as a carrier to various chemical substances, including metal nanoparticles and agrochemicals. For instance, microplastic can absorb pesticide, which can interfere with biosynthesis of gibberellins. When plants are exposed to this in the signaling pathways, their growth and development may be compromised, affecting crop yield and health plants. This is some challenge. The first is the accumulation in agricultural zone. The second is the second risk concern the potential contamination of crops. Studies shown that microplastic and nanoplastic can be found in crops, including common staples like wheat, rice, lettuce, and carrots. If these particles enter the food chain, they can have long-term environmental and health impacts, so particularly through the consumption contaminated produce. Finally, of course, we face regulatory challenges and the lack of awareness. Currently, there are not global standards for monitoring microplastic pollution. It's a pity, of course, in agricultural soil, and many farmers and consumers remain unaware of the potential risk posed by these particles. This gap in knowledge highlights the need for more resistance and clearer guidelines to address the growing issue of microplastic contamination in agriculture. In the slide I summarize, I revisit the key point regarding the benefits and risk associated with microplastic and nanoplastic in agricultural system. The risks are very significant, particularly concerning the accumulation in the environment. Microplastic and nanoplastic do not degrade easily, leading to persistent environmental pollution. And what else? This particle can also enter the food chain, and it's of course, therefore, is crucial between balance and this benefits and risk. Effective solution should be developed to minimize the negative impact, which is shown on the right column, negative impact of microplastic and nanoplastic, while capitalizing on the potential advantages. Of course, the future research and regulation will play a vital role in finding these balances. For Yep. As we look towards the future, there are several important solutions that can help address the challenges posed by microplastic and nanoplastic and agricultural. First, there is a need to development biodegradable plastic to replace traditional, for example, petroleum-based plastic. The second is improving filtration and removal technology is crucial, especially in wastewater and agricultural runoff. Another critical step is to regulate plastic use in agricultural, particularly reducing reliance on plastic-based mulches and fertilizers. And lastly, it is essential to promote public awareness and conduct more research on the long-term effects on nanoplastic, microplastic, nanoplastic pollution. Understanding, of course, this impact is a key to developing informed policies and solutions that protect ecosystem, human health, and agricultural system. Of course, another summary of challenges. One of the primary challenges is difficult in a large-scale production and extraction, for example, of phytohormones. Extraction is very easy in the large-scale production sometimes. These challenges are compounded by the instability and degradation of phytohormone, as they are highly sensitive to environmental factors such as light, temperature, and pH. Their rapid metabolism and breakdown in plant tissues complicate their use in biotechnology applications. Another challenge is the crosstalk between different phytohormones, which can result in unintended physiological effects. Meaning this interaction is key for precise hormone control in plant system. Analytical challenges also persist, so current detection methods require very high sensitivity and advances technical expertise. We know about LCMS MS techniques. Maybe another go. Oh, in sharp. Thank you very much. This is the last. Not the last. No, sorry, not the last.